This presentation is an introduction to the Rhesus or Rh blood group. I'm Justin Rhees from the University of Utah, Department of Pathology, Medical Laboratory Science program. After viewing this presentation, you will be able to describe the major Rhesus blood group antigens in terms of biochemical structure and inheritance, describe the characteristics of Rh antibodies, translate the 5 major Rh antigens, genotypes, and haplotypes from Fisher-Race to Wiener nomenclature, and state the purpose of Fisher-Race, Wiener, Rosenfield, and ISBT nomenclatures. And we will have additional learning objectives later on in this presentation. However, before we get to all that, I'd like to start off by telling you the very interesting story about how this blood group got its name. In 1937, a patient by the name of Mrs. Seno delivered a stillborn fetus at Bellevue Hospital in New York. And Bellevue Hospital is actually the oldest public hospital in the United States. Mrs. Seno had lost so much blood during the delivery that she required a blood transfusion. And her husband was actually ABO compatible. And as was frequently done in the 1930s, she received a transfusion of her husband's blood. However, a few minutes into this transfusion, she started experiencing chills with rigors, pain in her head and lips, and her urine was dark red. Mrs. Seno received subsequent ABO-compatible transfusions, not from her husband, rather from other donors, and survived. It was hypothesized that this acute hemolytic transfusion reaction was caused by an antibody specificity that was unknown at the time and unrelated to ABO. Such reactions after ABO-compatible transfusions had been observed in the early 20th century, but usually occurred in patients who had been previously transfused. Mrs. Seno had never received a transfusion before, and so it was postulated that she had produced an antibody against the fetus. In the late 1930s, a scientist by the name of Philip Levine tested her serum against 54 ABO-compatible blood samples, and only 13 were compatible. Around the same time, 2 other scientists, Carl Landsteiner, who discovered the ABO blood group in the early 20th century, and his colleague Alexander Wiener, were on the hunt to discover additional blood groups, and were immunizing rabbits and guinea pigs with the blood from the Rhesus macaque. It was presumed at the time that non-human primate blood contained the same or similar enough antigens to human blood. In 1940, Landsteiner and Wiener reported an antibody that was made by the rabbit and the guinea pig that reacted with about 85% of human red cell samples. And this antibody was called anti-Rh in honor of the Rhesus monkey. It was supposed that this anti-Rh antibody was the specificity that was causing these intragroup incompatibilities that were being observed. Equipped with both animal and human-derived antibody, it was now possible to type red cells for the Rh factor. And in 1941, Levine found in over 90% of erythroblastosis fetalis cases that the mother was Rh negative and the father was Rh positive. As time passed, however, it was recognized that the human anti-Rh and the animal anti-Rh were not the same. But because Rh was embedded into blood group antigen terminology, the name stuck. And the animal anti-Rh antibody was then renamed anti-LW in honor of the scientists Landsteiner and Wiener. The Rh antigens are proteins that are non-glycosylated and highly hydrophobic. They are transmembrane proteins that are thought to maintain structural integrity of the red cell membrane. And they may have a role in ammonia or CO2 transport. The Rh blood group system is a very large system that includes over 50 known antigens. In fact, to date, we're at about 61. It is one of the most highly polymorphic blood groups, which can lead to partial and/or weak expression of certain antigens. Of the many antigens in the system, the following 5 are the most clinically important in transfusion: D, big C, little c, big E, and little e. The RHD gene on chromosome 1 codes for the presence or the absence of the D protein. The D protein is what is known as the Rh factor. So when someone says they are Rh positive, that means they're positive for the D antigen. And when they are Rh negative, they are D negative. Most D negative phenotypes are the result of a complete deletion of the RHD gene, which is actually fairly common in certain populations, such as Caucasians, and is less common in African Americans, and is relatively rare in Asians. In close proximity to the RHD gene on chromosome 1 is the RHCE gene, which codes for the following combinations of the C and E polypeptides: big C little e, little c big E, little c little e, and big C big E. In contrast to deletions of the RHD gene being fairly common, deletions of the RHCE gene are exceedingly rare. And lastly, there's a third gene at play in Rh antigen expression, the RAG gene, or RHAG gene, which is located on chromosome 6. The RAG gene produces an Rh-associated glycoprotein, which is why it's named RAG, and serves as a co-expressor. The RAG gene, although located on a separate chromosome, must be present for successful expression of the Rh antigens. The gene itself does not express any Rh antigens. And we'll be talking a little bit more about this gene later on in the presentation. The D antigen is the most immunogenic antigen outside the ABO blood group system. In fact, as little as 0.1 mils of D positive blood in a D negative recipient is enough to elicit an immune response in the majority of cases. Next to the D antigen, the following Rh antigens are listed in order of immunogenicity: little c, big E, big C, and little e. However, anti-D and anti-big E are the most commonly encountered alloantibodies in the routine blood bank in the United States. So the question is, if little c is more immunogenic than big E, why is anti-big E antibody more commonly found? And the answer to this problem lies in this table of antigen frequencies of the Rh antigens found in Caucasians in the US. Although little c antigen is considered more immunogenic, it is found on approximately 80% of red cells. Therefore, only about 20% of recipients lack this antigen and could form an alloantibody against it. Big E, on the other hand, is found in only 30% of the population, and 70% of recipients have the potential of forming allo-anti-big E antibody if exposed to this antigen. This diagram shows the approximate time frame during which Rh antibodies develop after an initial exposure. And remember, because the vast majority of Rh antibodies are considered to be RBC immune, the exposure would occur through transfusion or pregnancy. After the initial exposure of the foreign Rh antigen, IgM antibodies are initially formed, followed by transition to the IgG isotype within about 2 to 3 weeks of initial exposure. As is the case with many alloantibodies, Rh antibodies can drop to a low titer, which could be undetected on an antibody screen, followed by a strong anamnestic response if the person is exposed a second time to the same sensitizing antigen. Just because a patient's antibody screen was once positive, it doesn't mean it always will be. For this reason, it's crucial that previously identified alloantibodies remain in the patient's permanent medical history. Most Rh antibodies are clinically significant and cause extravascular red cell destruction, severe transfusion reactions, and severe HDFN. Antigen-negative blood must be provided to any patient with a history of Rh antibody sensitization whether the antibody is currently reacting or not. However, complement-mediated intravascular hemolysis does not occur. And the reason for this is that Rh antibodies that sensitize to a red cell are physically too far apart to initiate the classical complement cascade, where at least 2 IgG antibodies are required within close proximity to each other to bridge the complement C1q complex and initiate the cascade. Because the complement cascade cannot be initiated, intravascular hemolysis is not seen. The antibodies, however, coat the cells and opsonize them, and then extravascular phagocytosis is facilitated, which can be brisk enough to cause harm to a patient, particularly in those whose anemia and other underlying conditions are serious. This table illustrates several characteristics of some of the most frequently encountered Rh antibodies. The optimal temperature for antibody sensitization is 37 degrees Celsius. The antibodies are primarily of the IgG isotype and can therefore cross the placenta and cause hemolytic disease of the fetus and newborn, or HDFN. And as previously explained, all begin as the IgM isotype and can occasionally have IgM components in varying percentages. All are capable of causing extravascular hemolytic transfusion reactions, although not all do. And the severity of the reaction depends on both the recipient's immune system, how much blood was transfused, and in some cases, the dose of the antigen on the donor cell. All are capable of causing HDFN in varying degrees, and dosage is variable. Although some examples of Rh antibodies demonstrate dosage, others, such as D, F, G, V, and VS, typically do not. And lastly, when routine blood bank enzymes physin, papain, trypsin, or bromelain are added, the antibody reactivity is expected to be enhanced. Okay, now for every blood bank student's greatest dream: learning the language and the different nomenclatures of the Rh blood group. The different nomenclatures serve different purposes. And we'll talk about the Fisher-Rhees nomenclature first, which is an attempt to describe both the genetics and the serology of the 5 most clinically important Rh antigens. Then we'll learn the Wiener nomenclature, which can be thought of as kind of a shorthand of Fisher-Rhees. Next is Rosenfield, which describes the presence or absence of the given antigens. No genetics are involved here. And lastly, ISBT, which is a system that catalogs the antigens within any blood group system. The Fisher-Rhees terminology is based on the theory of 3 closely linked sets of alleles: D, big C and little c, and big E and little e. And it's important to make a note here that the lowercase d is an amorph and does not produce a phenotypic product, where the lowercase c and e are actual antigens that can be expressed on the red cell. So little d will simply denote the absence of the D antigen. Because the Rhce genes are codominant, each inherited gene expresses its corresponding antigen on the red cell. While we know now that the Fisher-Rhees terminology isn't entirely correct, we still use it frequently in the blood bank, and so it's important to know. The Rhd and Rhce gene loci are so close together on chromosome 1 that they are inherited together on a haplotype. And remember that a haplotype is 1/2 of a gene. One is inherited from mother and one from father. This table illustrates the most common haplotypes in Caucasians and African Americans, as described in Fisher-Rhees nomenclature. From this table, you can see that the most common haplotype in Caucasians is the Rh positive combination D, big C, little e. And this is also the 3rd most common among African Americans. The 2nd most common haplotype in both Caucasians and African Americans is the Rh negative combination D little c little e. The fourth most common haplotype in Caucasians, this combination D little c little e, is actually the most common haplotype in African Americans. And it's just as important to know the rarer haplotypes as it is to know the most common. Note that the rarer haplotypes include Rh negative combinations where big C and or big E are present. And also, the Rh positive combination with big C and big E is present in less than 1%. If we look at genotypes in Caucasians, which, as you know, include two haplotypes, one inherited from each parent, the most common genotype in Caucasians is D big C little e, little d little c little e, which is found in roughly 1/3 of the population. The second most common is D big C little e, D big C little e, and the third most common is little d little c little e, little d little c little e. Remember that in Caucasians, about 85% carry the D antigen. Therefore, about 15% are Rh negative, and 15% are also little d little c little e, little d little c little e. This means that the vast majority of any random Rh negative unit from a Caucasian blood donor will be little d little c little e, little d little c little e. And the other Rh negative combinations, as you'll see on the next slide, are rare. This table illustrates the less common genotypes, most of which are found in less than 1% of the Caucasian population. Note that the rare genotypes include those that are Rh negative and carry big C and or big E. And I also want to point out here that the Rh positive combination D little c little e, little d little c little e, while found in approximately 2% of Caucasians, is fairly common in African Americans. It's found in about 20%. The Fisher-Rhees nomenclature is effective, but it's a lot to take in, and it certainly is difficult to express verbally. There was a need to simplify the information presented so that the haplotypes and the genotypes could be more easily expressed. The Wiener terminology can be thought of as kind of a shorthand version of Fisher-Rhees. Okay, as we learn this nomenclature system, let's first go over some basics. The presence of the D antigen will be denoted in Wiener by a capital R. The absence of the D antigen, which in Fisher-Rhees is indicated by a lowercase d, will be represented by a lowercase r. The presence of the big C antigen will be indicated by a 1 or a single prime. And the presence of the big E antigen will be indicated by a 2 or a double prime. Going a bit further, the Rh positive haplotype D little c little e will be denoted as RO. The Rh negative haplotype with the little c little e combination is simply denoted as little r. The Rh positive haplotype with the big C little e combination is R1, and the Rh negative haplotype with this big C little e combination is a little r single prime. The Rh positive haplotype with the little c big E combination is R2, and the Rh negative haplotype with this little c big E combination is little r double prime. So can you see a pattern emerging? Any guesses as to the next combinations? Okay, this is where things actually get a bit interesting. The Rh positive haplotype that carries both big C and big E is actually RZ rather than R3, which might have made a bit of sense. And the Rh negative haplotype that carries both big C and big E is little r little y rather than little r triple prime. Okay, an easier way to remember this is anything with a letter after the R, both C and E are big. One question I am frequently asked by blood bank students is, well, why do we need to memorize these nomenclatures? Why do we have to memorize the haplotypes and the genotypes? And truthfully, I really don't have a good answer for that, except that there are many times as a blood banker when you really do need to know this information. So here's a hypothetical situation. Let's say a trauma patient has come in, and you've had time to complete the ABO-RH, and the patient is A positive, and the antibody screen is positive. So you've run an initial antibody identification panel, and you're informed that the patient is being transported to the OR. While you are fairly sure that the patient has an anti-big E antibody, it matches the pattern, you haven't yet had time to complete the antibody workup. Run your selected cells or antigen type your patient, and suddenly the surgeon calls and wants blood for the patient's stat. What do you do? We know that blood should not be withheld from a patient in an emergent situation if it's required to save the patient's life. But the fact that the patient has antibodies could be a determining factor in the medical decisions the surgeon makes. It needs to be communicated that this patient has alloantibodies and therefore could have a hemolytic transfusion reaction to any blood transfused that's not cross-match compatible. And you need to inform to the surgeon that the antibody identification workup is still in progress, and that he or she must accept the risk involved in transfusing the blood that is not cross-matched. If the patient needs emergency release blood, however, what is the safest blood to dispense? Well, we know that approximately 70% of the random Caucasian donor population would be E antigen negative. However, from this previous table shown, we remember that among Caucasian blood donors, about 15% are Rh negative and about 15% are r. So if you had to give E antigen negative blood in an emergent situation, any Rh negative blood, which is ABO compatible of course, would be the safest blood to release. Rh negative blood that carries the big E antigen is very rare in both Caucasians and African American populations. And I'll show this again on the next slide. And just to illustrate one reason why we might use the shorthand version is, would you say I need 2 units of little d, little c, little e, little d, little c, little e stat? No, it's so much easier to say little r. This table shows that the less common genotypes that are Rh negative, which carry the big E antigen, are rare. And as you can see, once you've learned both of the nomenclatures, it's so much easier to visualize the genes and the antigens expressed once they're written side by side. So now that we've covered 2 of the nomenclatures, is anything missing? Yes. Both Fisher-Race and Wiener account for only 5 of the most clinically important antigens in the Rh system. And remember, to date, there are about 61. So as our knowledge and discovery of Rh antigens has expanded over the decades, it became necessary to come up with a more organized system to order them. The Rosenfield system is an alphanumeric system that starts with the blood group Rh and lists the antigens by number. If the antigen has been tested and is present, the number is listed. If the antigen is not present, a minus sign is written in front of the number. Rh antigens that are not tested are simply not listed. In Rosenfield, the D antigen is assigned the number 1. Big C is number 2, big E is number 3, little c is number 4, and little e is number 5. Additional antigens are also assigned numbers, but I haven't listed them here. In the example shown of an R1 little r person, in Rosenfield, this would be written RH colon 1, comma 2, comma minus 3, comma 4, comma 5. And this shows that the D, big C, little c, and little e antigens are present, but the E antigen, big E antigen, is not. So you can see that Rosenfield simply expresses whether antigens are present or absent. There is no attempt to describe the genetics involved. For example, which antigens are inherited on which haplotype, etc. Where the Rosenfield system is an alphanumeric system that's well suited to data processing, a system that's even better suited for this was developed by the Committee on Terminology for Red Cell Surface Antigens by the International Society of Blood Transfusion, or ISBT. In this system, which can be used for all the 35 recognized blood groups, the Rh blood group is assigned the prefix 004. Then, each antigen within the Rh blood group system is assigned its own unique number that completes a 6-digit number. For example, the big E antigen, as in Rosenfield, is also assigned the number 3. So, in ISBT, it would be written 004, meaning that it belongs to the Rh blood group, and then 003, meaning it is the big E antigen, or the third antigen listed in this blood group. One advantage ISBT has over Rosenfield is that rather than being alphanumeric, it is purely a numeric system. And it is now widely used because of its ease and adaptability to computer software. It is used also for all the 35 recognized blood groups, not just Rh. Okay, now that we've covered the 4 terminologies, let's take a look at some problem solving. So a patient's red blood cells are tested for the following Rh antigens: D, big C, big E, little c, and little e. And the antigens present are D, big C, little c, and little e. The antigen that is lacking is big E antigen. So can we take the serological results and, based on the most common genotypes that were shown in previous slides, make a prediction as to this patient's most probable genotype? Well, if the patient is Caucasian, the most likely genotype would be R1r. Because remember, this is the most common genotype inherited among Caucasians, and it fits the serological pattern. What are some of the other possibilities? Well, we have this combination, D little c little e, little d big C little e, or RO little r single prime. But remember that the little r single prime is a relatively rare haplotype. Now, if our patient is African American, this combination would actually be more likely, the R1RO. Remember that the RO haplotype is actually fairly common among African Americans. Based on serology alone, it's not possible to determine whether the patient is heterozygous or homozygous for the D antigen. And furthermore, we really wouldn't know the genotype unless we did the molecular testing. Question number 2. A patient possesses the following antigens, little c and little e. They're Rh negative, and they're negative for both big C and big E. So what is this patient's most likely genotype? Little r, little r. Are there any other possibilities with this one? Correct. No, there are not. So this was an easy one. And our last question, the antigens present on this patient's red cells are D, big C, and little e. So what is this patient's most likely genotype? The most likely genotype is going to be R1R1. And are there any other possibilities? And yes, this person could be heterozygous for the D antigen. Again, we would have to do molecular testing in order to confirm this. Here are the objectives for part 2 of our presentation. You'll be able to describe the D-, Rh null, and Rh mod phenotypes, compare and contrast the 3 mechanisms resulting in weak D phenotype, list 3 instances in which the weak D status of an individual may be determined, and 1 instance in which weak D status must be determined, and lastly, describe the following: G, F, CW, V, and VS. As we previously discussed, deletions of the RhD gene are fairly common. In contrast, deletions of Rhce are exceedingly rare. A phenotype that can result when the RHD gene is normal and yet there are abnormalities at the RHCE locus is known as D-. And this is an uncommon phenotype that lacks both big C and little c and big E and little e. And it results when the individual has normal RHD genes, and what would normally be at the RHCE locus is a hybrid of RHCE, RHD, RHCE. This is also sometimes called exalted D, because many examples of this have an unusually strong D antigen expression. And that makes sense because they have the D gene, and what would normally be at the RHCE locus, there's another RHD gene in there. The antibody that is often produced by these individuals is called anti-RH17 or anti-HRO, and it is essentially directed against the entire RHCE protein. If it is known that a potential recipient of blood has this rare phenotype, the person should be encouraged to donate blood for his or herself if possible, and the blood should be frozen to ensure the longest potential shelf life. Also, any siblings should be contacted because this RHCE mutation tends to run in families. There's a rare variation of this D-, phenotype, known simply as D. In this variation, the D antigen is not expressed quite as strongly as an exalted D, and the antigenic structure of the D. also carries a low incidence antigen known as Evans or RH37. And this variation was first identified in Welsh and Scottish families. Individuals with the Rh null or Rh deficiency syndrome lack all Rh proteins. And there are two known causes of this rare phenotype, the regulator and the amorphic type. In the regulator type, the RHD and RHCE genes are usually normal, but there is a mutation in the RAG gene. Remember, the function of the RAG gene is as a co-expressor, and it must be present for successful expression of the Rh antigens. So if you think of the RAG gene as being a regulator of the expression of these antigens, if the regulator is defective, no Rh antigens will be expressed. In the amorphic type, the RAG gene is normal, but there's a common deletion of RHD, so the person is Rh negative, and there are mutations in RHCE that silence the gene, leading to no protein expression. Individuals with Rh null syndrome demonstrate a mild compensated hemolytic anemia, reticulocytosis, and stomatocytosis, which supports the hypothesis that the Rh proteins have an important structural role in maintaining the integrity of the erythrocyte membrane. When transfusion is required, only Rh null blood can be given without risk of forming antibodies against any and all Rh proteins. And the antibody produced is known as anti-Rh29. The Rh-MOD syndrome results from a partial rather than a total suppression of Rh gene expressions. And this is due to certain mutations in the RAG gene. These individuals exhibit similar clinical features to Rh null, but the symptoms are generally less severe. The Rh null phenotype was first described in 1961 after it was found in an Aboriginal Australian woman. By the year 2010, Rh null had been identified in only about 43 people worldwide, making it even more rare than the Bombay phenotype, which to date has been identified in about 130 individuals. Of the 43 known individuals with the Rh null phenotype, it is thought that only a few regularly donate blood in Brazil, Japan, China, Ireland, and the United States. And the blood is therefore very precious. In fact, the American Rare Donor Program lists only about 12 donors. Blood banks will sometimes contact the ARDP to ask for Rh null blood to transfuse to some of the other rare phenotypes, such as D-, but it's really not a resource that can be used for that. It should be reserved only for Rh null recipients. In the fall of 2014, a fascinating article appeared in The Atlantic, describing a blood donor with Rh null phenotype named Thomas, who lives in Geneva, Switzerland. In Europe, although blood may be stored in a frozen state in countries such as France and the Netherlands, not every country has the capacity to store frozen blood in their blood banks. And it is extremely difficult, if not impossible, to transport blood internationally in a timely manner for a bleeding patient. When Thomas turned 18, he was encouraged to start donating blood for himself on a regular basis. However, because liquid blood has a short shelf life, his blood really should be frozen, and there is no frozen blood bank in Switzerland. So his blood has to be stored in the blood banks in either Paris or Amsterdam. In order to avoid the red tape involved with shipping his blood across the Swiss border into France, he will have to travel to Paris to donate. Soon after he started donating blood regularly, he received an urgent call asking if he would mind taking a taxi to the blood center in Geneva to donate for a newborn baby. Unfortunately for Thomas, because blood donors in Europe and in the United States are not allowed to be compensated for their donation, blood centers are also not allowed to pay for the donor's travel. And because of this, sometimes doctors will have to drive these rare donors to and from blood centers. Now let's take a look at the weak D phenotypes. Rh-positive red cells generally react strongly with anti-D reagents. However, certain individuals produce a weak D phenotype that can only be detected by carrying the testing through the anti-human globulin phase, using the indirect antiglobulin test. There are three known mechanisms for causing weak D: genetic, C-trans, and partial D, formerly known as D-mosaic. A weak D phenotype typically produces a reaction that is weaker than 2+, and most of them are negative at immediate spin. In the weak D test, anti-D antibodies will sensitize to the red cells during incubation, and then the tubes are washed, and anti-human globulin is added that binds to the sensitized antibodies and will form the visible agglutination reaction. Genetic weak D is caused by inheritance of D genes that code for weakened expression of the D antigen, resulting in mutations of the Rh polypeptide. Over 90% of weak D phenotypes are caused by genetic inheritance of single nucleotide mutations in the RhD gene that will lead to altered D antigen expression in the transmembrane regions or within the cell itself, rather than at the cell surface, where the immune system might recognize the alteration as being foreign. The D antigens that are expressed on the surface are therefore complete, but they're few in number. And inheritance of these genes can be tracked vertically from one generation to the next, and are seen most frequently in the African American population and are rarely seen in Caucasians. Because the D antigen is complete, these patients may be safely managed as Rh positive and may be transfused with Rh positive blood. In C-trans, the Rh antigen on the red cell is normal, but the steric arrangement of the big C antigen in relationship to the D antigen appears to interfere with the expression of the D antigen. And note that this does not occur when D is in cis position to big C. These patients also have a complete, although weakly expressed D antigen, and may safely be managed as Rh positive. The third category of weak D phenotype is partial D, formerly known as D-mosaic. And I've included here a picture of a mosaic with several tiles missing to help illustrate this category of weak D. If we imagine the D proteins as represented by this mosaic, the tiles missing will be certain epitopes of the protein. Partial D occurs when one or more of the D epitopes within the entire D protein is either missing and or is altered. Partial D individuals will test positive for weak D on the weak D test, but can produce allo-anti-D antibodies that will react with the epitopes that they're missing. Therefore, their antibodies will react with all D positive samples except their own. The allo-anti-D antibodies can cross the placenta and cause HDFN, and they can also cause transfusion reactions. So, once anti-D antibody is identified in a partial D person, only Rh negative blood should be transfused. There is no differentiation between genetic weak D, C-trans, or partial D in the routine blood bank. And policies regarding transfusion of weak D recipients are established individually within each transfusion service. Furthermore, regulatory agencies do not require routine testing for weak D in blood recipients unless the intended recipient has, or in the past has had, anti-D in his or her serum. Weak D testing is required, however, on all blood donors who initially appear to be Rh negative. A positive weak D test in a blood donor means that the unit needs to be labeled as Rh positive and should only be transfused to Rh positive recipients. Determining weak D status is important to discern candidates for Rh immune globulin, or RIG. RIG is a drug injected to prophylactically prevent true Rh negative individuals who are exposed to Rh positive red cells from developing anti-D antibody. So for example, when a mother is Rh negative and the newborn appears to be Rh negative through initial testing, it's important to determine the weak D status of the newborn by performing the weak D test to assess the likelihood of maternal sensitization and the need for RIG prophylaxis. It's recommended that all women who are Rh negative and who are not alloimmunized to the D antigen receive an injection of RIG at the beginning of the 3rd trimester and then again after birth. And other situations that might incur a fetal-maternal hemorrhage, such as a trauma, should also be considered. So in summary, the 3 situations in which weak D testing may be determined are for intended recipients of blood transfusion, for expectant mothers, and for neonates. And the scenario when weak D testing must be performed is on blood donors. Some recent updates to weak D testing. The CAP and AABB have recently recommended RhD genotyping be performed to determine the cause of weak D phenotype in patients. According to a 2014 survey of 3,100 laboratories, there is a lack of standardization of testing and interpretation of weak D. Some facilities routinely test for weak D on all intended recipients of transfusion, while others forego weak D testing on recipients altogether. And it raises an important question. Isn't it safer to forego weak D testing on all potential recipients of transfusion? Because by doing so, they'd all be managed as Rh negative and be given Rh negative blood, which is the safest thing to give. According to the workgroup's findings, by testing for weak D and performing genotyping to determine the cause, so whether the patient is genetic weak D or C-trans or partial D, this could prevent over 24,000 unnecessary RIG injections and could conserve over 47,000 Rh negative red cell units from being transfused in the US annually. Now that we've covered the 5 most important Rh antigens, the rare phenotypes, and weak D, let's spend some time covering some of the less frequently encountered Rh antibodies. And I'll comment that just because these antibodies are less frequently encountered, it does not mean that they are not clinically important in blood transfusion. The G antigen is a bit unlike the previously discussed antigens in that it is present on any cell, or virtually any cell, that carries either the D or the big C antigen, with rare exceptions. The G antigen is a separate antigen. So note, it's not a combination of D and big C, and that is a common misconception. This table illustrates the phenotypes that will most often carry the G antigen. So when D and big C are both present, or when big C is present in the absence of D, or when D is present in the absence of big C. G is usually absent when both D and big C are absent. One problem we run into is teasing out whether a patient has formed anti-G or anti-D and anti-big C, or a combination of all three. This antibody identification panel shows a pattern where the patient's serum has reacted with both D positive and big C positive cells. So how do we know that this patient has formed anti-D and anti-big C and not anti-G, or perhaps all three? Well, from a transfusion medicine perspective, it really doesn't matter. As you would transfuse someone with anti-G, the very same thing you transfuse if they had anti-D and anti-big C. For example, transfusing little r units that lack both D and big C would be appropriate. And if the patient has alloantibodies, a Coombs crossmatch would be performed, and units that appear compatible through the Coombs crossmatch would be acceptable for transfusion. So this would pick up any potential rare blood donors that are D negative, big C negative, and also G positive. Discerning anti-G from anti-D and anti-big C is a very labor-intensive process that can be done by immunohematology reference labs, and it includes adsorptions and elutions. Totally unrelated to the G antigen, the F antigen is present when little c and little e are on the same haplotype. In this example, both RORZ and R1R2 would show the same serologic results when we perform Rh phenotyping. However, only the RORZ example would react with an anti-F antibody because the little c and the little e are on the same haplotype for the RORZ, where they're not on the R1R2 example. It's important to note that F is not a compound antigen. It's a single entity. And because anti-F can cause both HDFN and transfusion reactions, it is considered clinically significant. CW is a low-incidence antigen that appears to be antithetical to the high-incidence antigen MAR. It's found in about 2% of Caucasians and is very rare in African Americans. And what's interesting about antibodies to CW is that there are examples of both RBC immune and non-RBC immune antibodies. In non-RBC immune examples, the person has formed the antibody to CW without prior pregnancy or blood transfusion, which is atypical of our Rh antibodies. Because CW is a low incidence antigen, it is very unlikely that a person will form this antibody, and even non-RBC immune examples are rare. Furthermore, it is unlikely that a patient will ever receive a subsequent transfusion of a donor unit positive for the CW antigen. So for these reasons, many blood banks do not require that this antibody specificity be routinely ruled out during antibody identification procedures. And last but not least are V and VS. These antigens are rare in Caucasians, but are found in approximately 30% of African Americans. So although relatively uncommon, antibodies to V and VS can be encountered in areas with higher numbers of African American blood donors. Also, most V-positive individuals are also positive for VS. And although the antibodies are considered to be clinically significant, finding compatible blood is not difficult. In summary, the Rh blood group system is clinically important in transfusion medicine because antibodies are capable of causing severe HTR and HDFN. Different nomenclature systems can be used to help describe phenotypes and genotypes. And because the Rh system is so polymorphic, antigens may be expressed weakly, and variations and deletions are sometimes seen. Although uncommon in routine blood banking, several other antibody specificities within the Rh system are of clinical importance, including G, F, CW, V, and VS. Here are the references for today's presentation, and I've also included the link to the article that appeared in The Atlantic, And thank you very much for your time and attention. I hope you learned something new about the Rh system today.